Okay, so as I mentioned, Nicolas joined the Genome Center in April 2012, where he oversees all the RNA-seq analysis, okay? He developed the pipelines for the RNA-seq analysis and supervises all the projects that are going on with this kind of data. He got his Ph.D. in computational biology in 2008 at the Institut Curie. Did I say that well?
Yep.
Agnes, what do you think? Not quite. But you understood me. She didn't understand. I knew it. Institut Curie.
Oh, Institut Curie.
Institut Curie. See it? I'm trying. He worked on yeast mitotic recombination and then did a postdoc over in Sloan Kettering in Eric Lay's lab, where he started with high throughput sequencing data of small RNAs. You all know about that. Now, we had a little lesson on how to isolate small RNAs yesterday.
Yep. We'll talk about it today as well.
And also talk about small RNAs. He just said that's great. So in addition to the discovery of dozens of new microRNAs, he identified a new class of piRNAs, which you also heard about yesterday, that map to the 3' UTRs of many genes of fly, frog, and mouse. In 2011, he joined this bioinformatics core, at Memorial Sloan Kettering, exactly, and then you moved on in 2012 off to Genome Center.
Yes.
So, as I said, he's here with us the whole day, and will start us on understanding what those FastQ files really are. All right.
Very good.
To Nicolas.
Okay,
Thanks for coming.
Thanks. So first, thanks Mona, Jason, the organizer, and I apologize for being late. I don't have any excuse for that. A few words. So I'm not a teacher, and I'm French, and I have a strong accent. So if you don't understand me, if you have any question, if you want me to repeat, feel free to interrupt me, and let's have it quite informal and more discussion. So I'll introduce myself a little bit more, and talk a little bit about the New York Genome Center, because you might be interested in what we're doing here. And then this morning we'll cover RNA-seq and really what we can do before mapping, before Top Hat. And this afternoon we'll go on after mapping and quantification of genes if you're interested in gene expression, and the kind of quality control we can do after mapping. So I started by doing an engineering college. I didn't do any biology for 5 years. I studied computer science, physics, thermodynamics, electronics, those kind of things, and that was very boring, and I didn't see myself working in an insurance or a bank like all my colleagues did. And it was about the time of the sequencing of the human genome, and people were starting to talk about bioinformatics. It sounds very interesting. So I went in Sweden to do a Master of Science in Bioinformatics, and I graduated from that in 2004, yeah. And went back to Paris, did a PhD in computational biology with 2 labs, one of bioinformatics and one of yeast genetics. And I studied the genome-wide location of meiotic double-strand breaks with microarrays at the time. And my PhD advisor was very interested in technology and was in charge of buying the first sequencer around 2007 for the Institut Curie. And so I was the single bioinformatician in his lab. We had a lot of discussion about this technology, and I realized that if I want to do a postdoc, I absolutely had to use that technology. And somebody was looking for a postdoc in New York. I realized it was a great opportunity to move to New York. So I moved to New York in 2008 with my daughter, who was 4 months old and just turned 6. And so I did a 3-year postdoc. We were part of the MOD-ENCODE consortium, which is the ENCODE. You all heard about the ENCODE, but the ENCODE for model organism. So for describing all the regulatory elements of Drosophila and C. elegans, and I was in the Drosophila group. And so we studied microRNA, endo-siRNA, and piRNA. And the class of piRNA that we discovered that mapped to 3' UTR, our paper was in fly primarily, but also mice and Xenopus. And there was a paper that I found yesterday that found it in macaque as well. So you see, that's very, very conserved. Although nobody understands what it does, and very, very few people are studying it. But still, that was interesting. So being at Sloan Kettering, it's also a great cancer institute, and it was an opportunity to do something else than Drosophila. And so I worked a little bit in the bioinformatics core, did our RNA-seq, so a longer RNA for a cancer project. And around that time, there was the announcement of the creation of something called the New York Genome Center, which I have heard before about, but I thought it would be a major sequencing factory that would essentially generate FastQ that the academic institution and hospitals will then process. And it turns out the announcement was that there will be more bioinformatics and there will be internal research. So I applied to this place, and it was really small at the time. So when I joined in April 2012, there was 10 people, right? And I was the second bioinformatician. The first one was hired the week before me. So what are we? So it's an independent, non-profit research institute. So it's not a company. It's a research institute. There is a lot of business people involved, and so there is a little bit of corporate culture, and it's really the key to turn a project from a 10 people into close to 100 people now. Yes. Yeah, sure, absolutely. Take pictures. So this is our building. So when I moved there in April 2012, we had only one office, and we could basically sit around a table. And then we opened a pilot lab at Rockefeller University. So we leased the space. We started to have sequencers there, and we had I think 5 HiSeq 2500. And we stayed there for 1 year, from July 2012 to July 2013. And the idea was really to bring together all New York institution, the academic institution, all together and create a big center of excellence for genomic. So for both genomic sequencing and bioinformatics. And so the plan was to be one of the largest genomic facility, and I think that's already one of the largest, integrating sequencing, bioinformatics, and big data management. And we'll talk about that a little bit. I don't think that number is adequate anymore, but it might be much over that. And the expectation is to create 500 jobs directly in the city. And so we're getting very close to 100, to our first 100 employee. Here are the list of the institutions. So from New York City, including Cold Spring Harbor, including the Jackson Lab somewhere here. And now we have 4 additional members that joined recently. So you see that all major academic institutions and hospitals in New York City are part of our organization. So we are an interinstitutional scientific community, a hub for relationship, and it's really like collaboration is really the key message here. That's exactly what we want to do, and that's just a first picture of our cafeteria, our big auditorium. We have 7th floor. We are located in 101 Avenue of the America, which is basically in Soho. So here is one lab. This is our terrace. And that's where I say that the view of the skyline here summarized very well our logo. So the logo was created much before we had this building, but it's a very, very nice terrace, which is Wi-Fi connected, so we can actually work there instead of and having lunch. One of the busy lab here. This is the building, so we have only the first 7th floor, right? And that's where the terrace is, right here. And it's located right here, so it's very well connected on the subway system, and it's fairly close to most of the local institution, and even Cold Spring Harbor is not that far, even though I'm late. We have 95 employees, so 30 are in sequencing in the lab, so technicians, lab manager, and sequencing project manager, and about 30 in informatics, and that splits between IT support, bioinformatics analyst, and bioinformatics scientist. We're actively hiring in all of these areas. And the product, yep.
Internships for students?
So every year we say we're not ready for that. So for this year, I guess that would be kind of too late.
Sure.
But definitely next year we will get interns. And yeah, we're
Paid internship?
that's probably something we can discuss. I mean, I don't know, but I don't mean, it's not something we have. We don't have a program for that. But I mean, that's really something we will seriously consider starting in September, and it will be paid internship.
Because living in New York is not cheap.
No, living in New York is crazy expensive, and we all know that. And without going into too much detail, that's how we attract people at the New York Genome Center. We have to differentiate between all the major prestigious institutions and our brand new institution. And one way we can attract people is also by paying them a little bit more. And we usually give them less benefits. We don't have housings, we don't have daycare, as I experienced and enjoyed at Sloan Kettering, but we can, we try to give a good salary, and definitely everybody who works at the Genome Center is paid, and there will not be unpaid internship. I think I can say that pretty certainly. Okay, so the product that we offer is so people contact us usually from the institutional founding members that I described, but also from any other places, any other institution in the country and internationally. And we have an initial consultation. We discuss the biological project. We discuss protocol. We discuss analysis plan. And that's usually sequencing project manager together with bioinformatics that are part of this consultation. We do the sequencing, we do the bioinformatics, and we store and analyze the data and store it for 2 years, although that number might change also. But the important part here is that we systematically do some bioinformatics, and that's how we can QC our data. We can control the quality of the data, and we don't want to release data that is not good quality. So people really contact us because they are sure that what they're going to receive is going to be high quality. There is no, if a library fails, if a library is contaminated, we will resequence that. And so the only way to make sure that the quality is good is to do bioinformatics, and that's something that we will cover today for you, because that is important at every step of any experiment involving high throughput sequencing. Numbers that are a bit off because it's really ramping up very quickly. We did around 500 whole genome, and I think the vast majority of them are human whole genome. Around 3,000 exomes, more than 800 RNA-seq, and again, I would say 85% of them are human RNA-seq, and the rest might be mostly mouse, and then a little bit of fly, zebrafish, mostly the very common mother organism. And I hope it's not too disappointing for you guys, but it's possible to do everything else. It's essentially the same technology, but it turns out the demand, at least in New York City, is really mostly around clinical application for sequencing, and very close to the clinic and close to the disease. So we do less exotic organism, but we can do it. And I think we were discussing last week the first de novo assembly that we will do. So an African bird for which we don't have a reference sequence, and we will do the sequencing and we'll do the assembly of that organism. We also do small RNA, microbiome, epigenomics, and those kinds of things. Here is our first lane or row of sequencers. We have 16 HiSeq 2500, and we announced in February that we will get in a set of HiSeq X10. So I don't know if you're familiar with this, but that's the latest product from Illumina that is shipped in 10 instruments that can only do human whole genome. And the 10 instruments cost $10 million, but they can do 18,000 whole genome per year. Okay, so each of them will produce. So one run takes 3 days and produce 15 whole genome at a 30x coverage. I don't know if that's familiar with you, but that's like the classical human genome that is done is 30x coverage, and so this instrument can do 5 a day. And we have 10 instruments, so that adds up very, very quickly. Yeah.
Why is, you said that they're only...
Yes, it's a purely artificial limitation for marketing purpose, and essentially for to cut the competition. The main competition for Illumina was the technology from Complete Genomics, who can only do human whole genome. And that's where they wanted to aggressively fight that market. It's a purely marketing strategy, and everybody believes that over time it will be lift, it will be modified. There is the technology, the chemistry is the same.
Yeah, because he actually the chemistry you explained to us the other day.
No. It's exactly the same. It's a limitation, and actually nobody really knows how it's going to be enforced. So it's not like they're going to align your reads and verify that you actually did a human whole genome. But on the other hand, nobody really want to try. When you have those instruments, you need to get the support, you need to get the reagent from Illumina. So nobody want to break the license, right? So we will take that instrument and do a whole genome, and we'll take all the other instruments to do the rest.
How much are you going to charge for, like, say, someone to have their genome sequenced?
So the marketing pitch was that it was the machine to do the $1,000 genome. It turns out it's really not adequate, and we are really trying to get under $2,000 per genome, and so there is a lot of there is no one fixed price. If you do large project, or if you want more or less bioinformatics, you'll get a different price. But it maybe 1,600 might be a reasonable price.
Is it really whole genome, or is it just the exome?
No, it's whole genome. Now, is that really a complete genome? That's a huge debate. And I think you talk with people from PacBio or that are using PacBio technology, and they will tell you that there's a lot of regions of the genome that are not covered by short-read technology, such as Illumina. So that's what Illumina calls the whole genome, right? The accessible genome with short-read technology. Okay, so a little bit about the hardware. So those machines produce a lot of data. We have a data center located in Pearl Street. So for those of you who know New York City, it's on the very close to the Brooklyn Bridge on the Manhattan side. We have a very fast connection between our genome center and that data center. We have 2,500 CPU cores, 9 petabytes of storage, and a backup and disaster recovery site in Quincy, Washington State, where electricity is much cheaper than in New York City. Just to give you an idea, this is 1 petabyte of storage. And on top of the Illumina instrument that we have, we also have an innovation lab where we can test new technology. Some companies are interested in placing their instruments into a genome center so that we can together develop their instruments. There is one called BioNanogenomics who does optical mapping, and so it can be very useful to identify translocation or gene duplicate, or genome or pieces of genome duplication. So we're working on that. Oxford Nanopore, you must have heard of that, and it's really ongoing. So we have received the instruments, and we're producing the first reads with that instrument actually today. And so we're very excited by this technology, who does really also long reads, and sort of compete with PacBio, although there's again a lot of debate around that. We also test new protocols. So there is this protocol called FFPE. I don't know if you're familiar with that, but that's really for formalin captured tumor sample who are stored in paraffin. And so there are a huge bank of those samples in every cancer centers, and it degrades a little bit the nucleotides, and so you need to get special protocol in order to sequence those samples. But we're working on that, as well as a lot of other places in the country, and we're getting close to have a very, very nice protocol to do genome and RNA-seq on those samples.
RNA-seq on those samples?
Yeah, we can do RNA-seq on those samples as well.
I mean, the RNA must be fairly trashed, though.
Yes, but there are, and I will describe that as a bioinformatician, so that's black magic to me. But they're working on it, and the data that I receive is, not as nice as fresh frozen tissue, but still it's usable, and you can have a very good correlation between if you take you do experiment and you do both, and at least with certain protocol for formalin capture, you can have a good correlation. So it may not apply to the entire data bank or biobank, but it's totally doable to do. So we work also on low input protocols. So when for various applications we have very small amount of samples, and up down to actually single cell genomics, where we can isolate a single cell and do either genomic or RNA-seq, and most probably RNA-seq. Sorry. Yeah, so it's really possible. There is now a lot of papers with that technology, and so you don't get the same range of expression, but you can still have very, very informative data with those technology. In addition, I just want to point that we also are recruiting faculty members Here are the first two members. So, well, we didn't get Jim Watson, who decided to stay here, but we get Tuuli Lapalainen, who started her lab in January, as well as Joe Pickrell. So they were both postdoc, and they're really starting their lab at the Genome Center, and they're co-appointed at Columbia University. And in January 2015, we'll have Yaniv Ehrlich, who is currently an investigator at the Whitehead Institute and did his Ph.D. Here at Cosmic Harbor in Greg Annan's group. Okay, so that's it about the introduction, about the New York Genome Center. If you have any other question now or later, I'm here the whole day, so I'm always happy to discuss about the Genome Center. As I said, I joined very early on, and there's been multiple phases, so I'm very attached to this project, and I can talk about it for hours. But for the next hour, we'll talk about RNA-seq. And so I was not here the previous days. I'm not sure what you did, what you know. I met with Michael Ronan, who you met from Illumina, and he was at the Genome Center yesterday. So he told me you were a fantastic group of students and that you knew everything about it. So I realized that my presentation, that's what he said. That's what he said. That's what he said. And so then I realized that my presentation might be too basic, but we'll see. We'll go and tell me, and we'll adapt. Very good. Okay, he was very impressed anyway. Okay, so I tried here to just make a small description. What we're talking about here, we're talking about a fragment of cDNA here. So the RNA is fragmented, and we get this fragment. And what we're going to sequence are the extremity of that, okay? And so we're going to get a lot of them, but what we're going to sequence is the red arrows. So we do the cDNA fragment being sequenced, and it produces FastQ files. So what goes here between sequencing and FastQ file, I don't know how that has been described, but so there is imaging. So the sequencing, I'm not going to describe the technology of sequencing. But basically, for each position in the read, there is imaging, and we image progressively the 4 different wavelengths that correspond to 4 different nucleotides. Okay, so you have one slide, and there is a very, very, very high definition camera that take a picture and identify the level of excitation for each position on the slide, corresponding to each of the bases. Okay, so basically it say, do I have an A, do I have a C, do I have a T or a G at each of the location, right? But so the raw data at that level, at least, the raw data are image, right? And so after those, we need to transform those image into binary file or quantification file. And so there will be a measure of intensity for each location, so for each pixel, corresponding to the 4 wavelengths, so the 4 nucleotides, on each location on the slide. Okay, and so those files are called BCL files. They correspond to intensities for each location. And so then there is some image processing to identify cluster, and so group of pixels that are correspond to the same read. Okay, I was not planning to talk about that, so feel free to interrupt me if I'm confusing. But those ones are BCL files. They really correspond to the intensity at each location, at each of the cluster on the slide. Okay, and the process here that we call base calling is to transform those intensities into sequences. Okay, so basically you have I'll need this, or maybe I won't. So by as a parameter of your experiment that you expect 100 nucleotide on your reads. Okay, the reads are 100 nucleotide, let's say, and so you have 100 files, that describe the intensity of those 4 colors for each location. And so for each file, so for each position in the read, and for each location, you will call if that's an A, a C, a T, or a G, okay? Which is the most likely intensity at that location. Does that make sense? Mm-hmm. Okay. And so the process of calling those bases goes with a quality score. So we call the base, we say it's an A, but we also give a quality score that say, I'm super confident it's an A, or I'm not so sure and it might also be something else, okay? Or, you know, it's like not always at the same location and that cluster might not be so great, right? And so what we end up with is the FastQ file. So I'll show you an example after. FastQ file, so it really comes from FastA that was actually, first of all, it was an alignment algorithm from the 80s, which is like prehistorical science from my point of view, not my point of view, my experience. And so then, you know, the legacy of that algorithm is actually the format that it was using. And FastA files are widely used, and they contain a header that describes the sequence and the sequence. And FastQ files are an extension of that contain a header, a sequence, and the quality score that corresponds to each base, okay? So the confidence that we have in the calling of each of the base. And so there is 3 elements, a header, a sequence, and a quality score, and because it's very logical, that makes 4 lines. And so the first line always starts with this R base here, and that's the header here, the seq ID, then a sequence, and then there is this plus here that optionally can contain also the header. So sometimes, you know, you would have the header present twice, and then you have the quality score here, okay? And that's kind of like an historical format. There's absolutely no reason to have this 3rd line here, but it was there, and it's still there, and it will be there forever, okay? Because essentially all the algorithms that have been built on this format will expect to have this, okay? And so it's too late. Either we change the format or we keep it that way. So the first line starts always with an A, this arrays, and contains the header. The second line is the sequence. The third line is nothing, but it starts with a plus, and the quality score of the sequence. Yep.
Why is there a third line?
Uh,
That's fine.
Did it once have a reason?
Is it to
So-
indicate the end of the sequence or something?
No, because it's a different line. So no. It used to. So there are so many files where it had the header again. Okay. I don't think I ever saw something else than either the header or nothing. And the historical reasons, I'm not sure. One bad reason is that it's kind of easy to say that the sequence are always the second line and I don't know.
Is it because it comes from FastA and maybe?
Yeah, it just is some. I mean,
The right person did it and it's stuck and you're never getting rid of it.
So it's a legacy.
it's a legacy. Yeah. Yeah, so that's a good point. So of course, you can all read this the sequence, and that's very, very familiar. This is not familiar, okay? This quality score here is not familiar, and so it's supposed to represent the probability that the base call at the location is incorrect, okay? So essentially, the confidence that we have in the calling. And it's encoded in these ASCII characters. There is 94 values from 0 to 93. And here I copy and paste from Wikipedia from the low quality to the high quality, okay? And so I cannot read that, but on Wikipedia you can find easy parsers that will convert this string into values, and those values are usually between 0 and 40, or they could be between 0 and 94 actually. But in the current implementation, it goes really mostly from 0 to 40. So yeah, there has been different version of that. I don't want to go too much into what are the different versions and how they differ. There is now sort of a standard that it's an Illumina standard, so that's what we're going to use here, and it really goes from 0 to 40, with 0 being I'm absolutely sure it's a wrong base, okay, to 40 that says I'm fairly certain that it's the right base, okay, and so if we go back here, so one problem that we have is that we have tens of millions of those entries, okay? So here I just represented one read, and imagine we have 10 million or 100 million of them, and we want to compress that, okay? So this is essentially, I used to work in computer science, and essentially this is a text, and we want to compress that, and we don't know anything about the format of this file, but we know that here, if we knew anything about the format, we would know that here there is 4 different possibilities, okay, and here there is 94 possibilities, and we can take advantage of that to do a more efficient compressing, okay. So if you know that there is only four different possibilities, you can store that information on a small piece of memory, okay? And you can then compress and say that here there is 3 A's, and you can compress that in a more efficient way than storing A, okay? And here, if there are, again, if there are characters that are successively the same, you can compress that efficiently. But so one tendency at the moment is to reduce the number of bins. So instead of having 0 to 40, reduce that into maybe 8 or 16 values. And so there are, that would make the compression much more efficient. That would reduce the size of the compressed FastQ files. And so people are arguing if that will have an impact on the downstream analysis. Will we get less efficient, or is that something we can afford and still do the analysis the way we usually do without losing too much information? So is compressing the likelihood that we give to each base might decrease the efficiency of some of the tools, but it might also lead to better compression, okay?
Would that mean all the algorithms would have to be redone if you did that?
Not necessarily. So for RNA-seq, we do, or so for gene expression with RNA-seq, we do use the quality score in the alignment. And so, so we'll describe the alignment later today, but when you do an alignment, when you have a mismatch, if the mismatch is at a base that has a low confidence, it doesn't matter as much as if it's a base with a very high confidence. And so you calculate the overall quality of your alignments. And so if you change the quality score scheme or scale, there might be a read that suddenly will not map. But that would be very, very marginal effect, right? That would be a very, very small difference.
So you wouldn't change the alphabet. You just have less in between.
Yes, exactly, right. But where it matters the most is for variant calling. So when we do exome sequencing or genome sequencing, the most important thing is to see where the sample differs from the reference sequence, and that's where quality score are extremely important. Okay.
That's what I was wondering is how do you correlate, whatever score if it's a 20, how do you kind of correlate to whatever variant such that relative to some allele frequency,
Yeah. So when they do variant calling, again they give more weight to a base that has a high quality score, and it's different from the reference. Okay. So we're quite sure that at that location we have something a mutation. Okay. If the quality score is low, then we have less weight. And if all the you never do that with one single read. So you do like 30 reads, and if all the base that points you to mutations are of a low quality, then that's problematic. You know that it might not be a mutation, Okay, it might be just something else, a sequencing error that happens always at the same location, or well, it's multiple scenarios that can lead to that. But if you have strong, a very good quality score for all the bays that support a mutation at that location, then you're confident that there is a mutation. There is a difference between your sample and the reference. Does that make sense? Yep. Okay. Cool. Okay. So two things, or a few things about the FastQ file. The line 4 and the line 2 and the line 4, they must absolutely have the same number of characters. Line 4 is the quality score for each of the bays, and so it's most of the algorithm will break if those two lines don't have the same number of characters. Yes, the R base and the plus, they're possible quality values. Okay, so if you have a plus here, that's a valid quality score. So that means that if you want to count the number of reads, you shouldn't just count the number of at here in your file. Okay, that won't give you the right number. You have to count the number of reads in a different way, and we can talk about it. The extensions of the FastQ files are frequently .fastq, .fq, and the first line in the Illumina sequencing can contain some important information. So this is just an example, and so that was a dummy example here, but here is a real example where the first elements, so all the elements are separated by the semicolon. The first one gives the instrument ID, and then the run ID, the flow cell ID, the lane in the flow cell, the number within that flow cell, and the coordinate on the flow cell on the slide. Okay, and so those information can be useful in post-processing. I'm not sure what the Next-seq FastQ files look like, but I suspect they should be quite similar.
I think it's about the same. I
Yeah.
don't know if they've been the quality of Next-seq.
I don't. Okay, cool.
And
I saw
I'm not
just
sure I understand the number within that percent. Is that the number for the cluster or?
Let me see.
Don't worry.
I'm actually not sure. I realize now.
Because then, I mean, X
Yeah.
and Y coordinates will seem to be
Redundant.
precise, then will be sort of redundant to flow cell lane and.
So
within the flow cell.
it might be the tile. So flow cell is separated into squares. Well, lanes, but within a lane you have the tile,
Okay.
and it might be that number. And yeah, I'm not.
So let me just be clear.
Yep.
So these values and the information, as well as the derived sequence, are generated in the Illumina instrument, or is it the I'm sorry.
Yeah.
That was probably covered in a couple of.
Again.
No, good question.
So what's the output of the Next-seq?
Well,
Is it the?
I'm just wondering, where does all this information get added? I understand
That's-
that the Illumina does all the imaging,
Right.
right? And
So-
so the image files are then processed on the Illumina unit?
Yes.
Or that data is?
So usually yes, on the Illumina unit by a set of software called Cassava. I don't know if that was mentioned, but that does, so that process is called BCL to FastQ, okay? And so the BCL are the quality score files that I described, and the FastQs are those one. And so that's during that process that this information is added, okay?
So is number within the flow cell just like the cluster number?
Yeah, it might be, but I'm not quite sure if that's the number or the tile number.
I guess the bottom line is, when do we have to care about this information?
Well, we'll come to that.
you have to care about how it gets there, and he's going to show you just one or two things to look out for.
But once in a while you have to care about it. Ideally, if everything goes well, you don't. Okay? But contrary to what,
I don't know. For me, I have like really curious students.
yeah.
So if I don't know what things are, it's like, well,
Yeah.
it's important, but I don't know what it is.
And you're always going to
Yeah.
have the one student who don't want to know what every single number means, so it's nice
Tough.
for us to
Yeah.
have
Suffer through it.
The
Yeah.
point them.
Yeah, tomorrow at Woodbury, Elena, who actually is Cornell's wife, is going to give a very brief description of what happens when when she gets those raw data files and what she sees, and is able to transfer them and deconvolute and demultiplex them, she'll go very briefly and tell you what the BCL file is.
Yep.
That's tomorrow.
Okay. Cool.
That would vary.
So yeah, I didn't mention this demultiplex, but you'll talk about that tomorrow. Okay. Okay. So single end, we have one single FastQ file, and paired end, two files. Usually they contain R1 and R2 in the files. I saw this morning from your files that that's the case. And those two files, they must absolutely have the same number of reads. And the paired reads usually have the same header, and that's how we can find them. So now we know what is the file and what kind of quality control. The first thing that I would suggest you to do systematically is to just look at the files, okay, and open them. And so, but be careful when you open them because they have millions of files, millions of lines. So if you do, so that's a question I have for you. Is the plan of the course to teach you how to use DNA subway, or will you use the file? Will you do this process differently?
We can actually have now that people have data, we can, and some people who did not have data have already done a FastQ dump from some SRA files.
Okay.
So we can go and show them. We can open up the file. DNA subway does not do that.
Okay.
But we can open up the file in the discovery environment, and that's why somebody asked me when they saw, their file. I was like, "That's it." And I said, "That's page 1 of 22,000." But later, maybe tomorrow or later on what he's showing you there, we can do that
Yeah.
command line later.
Yeah.
Yeah.
So here are just a few comments. So if you do cat of the file, that's not a good idea because that will just try to output every single lines in the file, and that's usually way too much. What you can do is head of the file, and that will give you just the first 10 lines. And if you do head dash a number, say 100, that will give you the first 100 lines of the file. If you do less, show the file page by page, okay, and you can then navigate in the file. And maybe it's very familiar for all of you, but just I think it's useful to just look at the file. And I'm guilty of not doing it systematically, but I'm also processing hundreds of files at a time. So this is really, there are cases where you do a lot of processing of the file, and you could have catch a problem just by looking at the file, okay? And so if the file is compressed, you can do zcat of the file with the GZ extension, and then head or less, and that will give you the same result, okay? I think it's important to just look at the first lines, because sometimes like all the reads will be identical, or there will be no reads, or just look at them, make sure that they're the right size, like huge size. That's usually a good indicator. If they're really tiny, there's a problem, and that they really contain something in there, okay? Now, if there is something bizarre in the very first line of your file, that can be fine, because usually the files are ordered by their location on the slide, and the very first few reads, they might be in a corner of the slide, of the lane, and that's usually not very good quality. So, go through a number of lines to make sure that it looks okay.
You just get all these weird symbols.
What's that? Did you just look or?
I just opened up the discovery environment.
And
Oh, okay.
it doesn't look like a FastQ file from here. It looks
No,
like a zip
it's-
FastQ file.
Yeah.
Okay.
So I don't know if there's a way to.
So, they're zipped or they're
Yeah.
GZ?
Yeah.
Okay, well.
That's what I can see from here.
Okay.
But
So
that
we'll do
we'll
it.
get into later on.
Okay, we'll do it. And I'll do it with you. Okay, so the other thing that is very widely used, probably everywhere at every genome center, is this thing called FastQC, and that's part of your DNA subway environment. It's a standalone software. It needs to be installed on your computer. It's a Java application. Again, that's when you want to use it yourself on your computer. Otherwise, you can use the DNA subway version. And it computes a set of metrics just from the FastQ file. And what I will do now is on the FastQC website, there is an example of a good run and a bad run. And so we can go together through all of those metrics and discuss, what they are and what do they represent. Here, the first thing that you will do systematically everywhere, the very first QC that everybody does is to check the quality along the sequence. Okay, so here it's a bar plot, or it's a box plot histogram that represents at each location in the read what is the mean and first and third quartile of the distribution of the quality score for that location, right? So here, the first location, the mean quality score is 37, okay? So on a scale from 0 to 40, right? So it's very good. And then you see that it stays very good. It's even better around between 5 and 10. It's very, very consistently good until 32, and then it slightly decreases at the end of the read. And because of the technology, we know that the chemistry is not as efficient at the end of the reads. The reagents basically get tired, and the cluster just fall apart. And so there is more bad quality, so more case where we cannot call efficiently the base at the end of the reads than at the beginning of the reads. So you will kind of systematically have a decreasing quality score, okay? But overall, this is a very good read where it really stays. So they indicate here in green, like some threshold that shouldn't be taken too literally, but this is a very, very good quality. What we want to see is that it's consistent. It doesn't drop too dramatically, and there is no like one location that's completely off. Okay, once in a while you have a trend, and suddenly you have one location that's completely down. Okay, and so.
Is there any straightforward translation or conversion between the Q scores and the percentage that it's that it's not wrong?
Yes. You're asking.
Yeah.
What's the
What's the
value?
fraction?
What's the probability?
Yeah.
Of
Like
Well,
percent.
a score of 37 says that I'm 98.9% confident.
It's the definition of fraud.
So it's something like that. There is a conversion into. Let me see.
Percentage
It might be.
It's not a so there is a log scale that's involved. Okay, and so I'm trying to. Yeah, I'll give you that.
I believe it's 20 is 1 in 100 error, 30 is 1 in 1,000.
Yeah.
40 is 1 in
10,000.
Yeah,
So-
it's actually right in the Wikipedia page.
Okay, the
so
Wikipedia page tells
A
you.
score of 30 is exactly what we said, 1 in 1,000. A score of 40 would be 1 in 10,000. So a
So
score of 30, it's
The odds, it's
99.9%.
1 in 1,000.
one error out of 10,000. Okay.
Yeah, probably the incorrect base is 1 in 1,000. So the base call accuracy would be 99.9% of a FRED of 30. Yeah.
Perfect.
And it's just 10, 20, 30.
Yep.
It's 10 raised to that number.
Okay. Does that answer? Okay, cool. Thanks. Okay, so bad quality now. So you see exactly what I described. It's dropping dramatically, and starting at position 20, you start to have very large number of reads that have low quality. Okay, so this is problematic.
Where is that information about the number of reads that are of low quality?
So there's no information here about the number of reads. So that's what we see here is the distribution of the quality score per location. Okay, so the red line here is the mean. Okay,
Sorry, the red line is
is the mean quality score
okay.
at this position, and I guess here is the first quartile and the third quartile.
So the yellow is the first quartile.
Yes.
Okay.
Okay. So
Throw this away and just say that's not good.
Well-
They need to resequence everything?
Well, that one,
it's so the quality score, the quality control operation is to compute a set of metrics that will inform you on the decision that you have to make. And I can't tell you like, no, this is bad and you have to resequence. Sometimes there might be reasons to explain why we obtain that, and maybe that's expected. And sometimes it just you cannot resequence. You just don't have any more sample. You don't have the money to do it, and so you have to deal with that. Okay. So there is no strict decision to be made. It's really on a case by case. That's the stats that we use to make those decisions. Okay, so the other one, and I believe this is on a new version of FastQC, so I don't know if that's implemented in the DNA subway version, but essentially that's the tide, the quality per tide. So here again we have the position of the read, and we have the quality that are colored. So this is just the location on the slide, and we want to see if we have an homogeneous quality score. So if overall all the location on the slides are of equal quality, or if there are locations where suddenly geographically on the slide there is a bad quality score. Okay, so that's what we see here for location 2. We see here and here there are really locations where the quality dropped. Okay. So, and to go back to one of the original questions, in order to get this slide here, you need to get the position on the FastQ file. Okay, that's the information you will use to make the slide that I just showed. Okay, so that's one example, and there will be another example this afternoon if I remember. Now the quality score, so that's just an histogram of the quality score. You see here that the vast majority are above 30, and you see here that you have a lot of quality score that are below 20. Okay, so that's pretty much the same information that you had 2 slides ago, but that's another representation. Okay, it's not about the location on the read. It's just overall you have either a vast majority of the base that are good quality, or you have a lot of them that are not very high quality. The base composition, so at each location on the reads, how many A's, how many C, how many G, how many T's do you have? And you don't expect necessarily to have 25% of each, because you can have bias in the organism or in the transcriptome of your organism, but you expect to have somewhat homogeneous, somewhat constant value for this. Okay, so here the good quality, it's a straight line, and here the bad quality, you have the very first location, you have more of I believe it's more A's, and here you have at that location you see that you have one of the base G's that is much higher than in the previous positions. So you have a lot of along your reads, you suddenly have a peak of G's, and biologically there's no reasons why position 22 in your reads should have more G's than position 20 or 18, right?
So what would be the reason for that? Like a technical problem?
Yes.
At that time during when that base was being added, or?
Yeah, so that could be a technical problem during the sequencing. Okay, and so say there was 2 more of the reagents, of the G reagents at that location, or too few of the other ones. That could be also because if you don't have a good diversity of your sample, you have a read that has a G here at that location, and that's super highly abundant. Okay.
Because that's what I have online.
Yeah. So
This is awful, isn't it?
this is very interesting.
This is actually not my data. This
Okay.
is actually an SRA from NCBI.
Oh, okay. Well, so give the names. Not all sequencing data is super high quality, but so wait. So
A lot of ours failed that.
so is that a small RNA sample or
No,
it's
it's
okay?
a regular.
Yeah.
It's an RNA-seq.
Yeah.
Still.
Yeah.
Do you know how long those reads are?
49.
29.
49. Yeah. Yeah.
I'm
Well-
just curious.
There's a lot of ours.
Sorry.
Yeah, a lot of ours failed there, like the first 9 were really noisy. and
Okay.
then
Yeah.
totally
So is that also SRA or?
No, this is
Okay.
We'll
So
talk about that when we get into
Okay.
yeah.
how to
There's okay. I didn't realize you were all looking at your files at the same time, but that's great. That's perfect. Perfect. So I don't want to say anything bad. I mean, all your quality is wonderful.
We are intent.
That's fine. That's fine. That's fine.
So I actually kind of wanted a little clarification of Agnes's question. So it's technical. I'm trying to figure out technical at what level? The machine, the loading, on the machine.
So, I mean, I guess it could be pretty much every level. You can introduce some problem. The situation that we see here. So all of the ends of the reads are all the same essentially, and you can basically find what it is. So the end of the reads are all like A, C, T, whatever. And you could have that if all of the reads that you were interested in where there is diversity, but they're all like 20 nucleotide, and then you sequence the adapter sequence. So at the end of your reads, you had adapter sequence that are all the same sequence, and usually you don't sequence that. Okay, they're after your fragment of interest. But if your fragment of interest is too short, say 20 nucleotide or say 21 nucleotide for microRNA, then you start to sequence systematically the adapter at the same location. So position 22 will be the first base of your adapter, and 23 would be the second base, and 23, right? And so if a large majority of your sample is 21 nucleotide, then all of those reads will have the adapter sequence from position 22 to position 50. That makes sense? And so here, you resequence always the same thing. At the location you have, every time you have the same location. The problem for that we had in the 5' end of the reads, I'll have to think about it. Yeah, I don't know yet, but yeah.
Are these really, position in the read, position 1?
Yes.
The GC content.
This is, so these are all reads that start at that position and go
Yeah.
third.
Yeah.
One fails.
But there are, that same base position is covered by a different read that began 5 nucleotides upstream.
Yeah, but yeah, sure. But you would only know that after mapping. Okay? Here you don't have any mapping information. It's just FastQ file. Okay? That's all you have. That's the only input for that program. And so they're completely independent sequences.
Is there some type of cutoff within which you use for map once to proceed to the mapping step that you would or would not use for mapping?
Well, so to be honest,
Use everything.
I'm not even sure. We usually have that information plus a lot more metrics. And so when we have a doubt, we will kind of know it in advance. So the Illumina instruments also output some quality during the run. And so if there is anything problematic, we would see it and we would run FastQ and then make a decision. But usually at the Genome Center, we systematically run the alignment. And I'll describe that this afternoon, but the alignment is actually fairly fast in our hand. And so there is an advantage at doing it automatically without any human intervention, rather than waiting and taking decision for each sample in our setting, okay? In other settings, you might want to do, so as you saw, FastQC gives you like 3 indication if it's okay with a green light, or if it's a warning, or if it's an error. And those there could be an indication. They are not strict thresholds, and there is no strict thresholds. I mean, I think it's really based on experience that we know in advance what is going to be a problem or what will be fine.
I guess
And
I'm non-model work.
Exactly. So that's
Kind of problematic.
it is problematic, but it's also why I want to give you a threshold, because the threshold that we have for our human samples when we have enough material might not apply to other experiments, right? One thing that's for sure is that if you want to do assembly, so even transcriptome assembly, you really need to get higher quality data, okay? Having not ideal quality for an experiment when you want to do gene counting, it's actually it can be fine. And you have to see that if a read is really bad, it will not map to the genome, right? If it's really, multiple errors along the read, it just won't map, okay? There's no chance that it would map by accident to somewhere else. So in a way, that's a way to filter out the very low quality reads. Now making a decision on the entire library, well, some are obviously very wrong, some are obviously very right, and there is a huge gray zone in the middle where you have to make a decision, and we'll go through your guys' data, and I'll try to give you indications. But yeah, it's mostly an experience thing, I guess. The other metrics that's computed, and you can also look at, is the GC distribution. And so the red here is, say, the GC count per read, and the blue is the theoretical distribution that, as far as I know, is tuned to human genomics. And I'm not even sure, or like human genome, and I'm not even sure that's adequate for transcriptome. Okay, so the sequence.
So even an iPlant, it would be an iPlant. Oh,
so
because they're running the FastQC. Got it.
Yeah, so they're running the FastQC, and you'll have this theoretical distribution, and that's sort of a normal distribution around 50, right? But some of your organism might have a bias, or might
Right, may as well.
Yeah.
Yeah.
And so if you don't fit that theoretical distribution, that might be fine. But yeah, that's organism dependent essentially.
I have an exclamation point in that one.
Sorry?
I have an exclamation point in that one.
So that's the orange. That's the warning. Yeah, I think that's pretty common.
Okay.
And I wouldn't worry too much at this stage on that. I mean, if that's the only warning, then you're all fine.
Well, I have plenty of red.
Yeah, we'll go through that. That's on the SRA sample or on your sample?
Yeah, it's still the RNA sample.
Yeah, but apparently it's, it's very. It seems very problematic, that one. I hope it's not one of my data. So.
What happens if there's just the red line? No, the other.
For which? For the GC?
Only red line there. Where?
Oh, there is no blue line?
What's that?
You don't have the theoretical distribution? So
Sequence GC content.
you guys have it, or
Yeah,
just
For the per base GC, I only have like
you only have the red as well?
the per base.
No,
and
oh,
then
yeah,
it's
yeah.
this one, and then
Oh,
there's
that's the
sequence.
yeah. So you're I didn't put all of the graphs. So the one here is the per sequence GC content.
Yeah.
I can show you, and I think it should have the blue line. Yeah. So if you go to the below that one. Yeah, here it is. Okay.
So what's that? The other one mean?
Okay. So the other one, let me see.
What's this?
Yeah, so the graph that is right before this one is the GC content along the read. Okay, so again, per location, do you have an homogeneous amount of GNC versus A and T, or is there, more ATs at the beginning of the read than at the end of the read? Okay, so that's the same value that is computed here, but along the read, and you expect to have a very homogeneous, a flat line, and not a bumpy line or a any other sort of line. You want a flat line. Okay, so the duplication level. So that's how many reads are identical. Okay, and so you don't want too many identical reads because you want a diversity of reads. But usually you have a step of PCR amplification in your protocol. And so you can have one molecule that goes into the sample preparation that gets duplicated. And so that's something to be worried about. If you have too much identical reads, that might be that a significant portion of your library is resulting from a PCR artifact and is not reflecting the original sample that you try to sequence. And that will affect the estimation of the gene expression, okay? Because for one gene, you started with 1,000 fragments, and they get highly amplified, and you get 10,000 fragments that are sequenced, then the expression of that gene will be biased, okay, will be overestimated. So here is a good example where the vast majority, like close to 100% of the library is unique, and a little bit of the reads are present in up to 10 times. Here is a bad example where you have a lot of reads that are present up to, I think it's 5,000 times here, okay? So that can be problematic, okay? So you want really a continuous distribution here.
So my plot looks like a U.
Minus one.
Yeah.
What is that?
So with like a lot of them at the 10K?
Yeah, yeah.
Well, not 10K, ours are
Yeah,
10
Yeah, so
plus.
9, 10K, yeah.
10 plus.
10.
Like I think our x-axis is
Oh.
goes 1 to 10. It doesn't say K.
I'm missing the blue line.
Oh,
Yeah,
it's stopped here?
Yeah, ours just goes from 1
Okay.
to 10,
Okay.
and there's no
Missing
blue.
the blue line.
Ours says it's the duplicates relative to unique. I can't quite read what your red is.
Yeah.
Mine is deduplicated sequence and total sequence.
I think ours is just showing ratio.
Can I have a look?
Yeah.
Yeah, I think we all have the same.
Yeah, because we're all DNA subway, we're all gonna have the same.
Okay.
And it gives you a fail, but I'm not that worried about where
Yeah.
it falls. Yeah.
Okay. I mean, I think so. We'll go through that. I may need to know a little bit more about the samples,
Okay.
about. So yeah. So again, there is different version of the FastQC that.
It may actually be a different version because they're all using the same version and there's no blue line on these.
Yeah. No. So but so I just want to know if the sum of all these numbers just make up a bump at the 10, and so that might just be normal.
We gave it a fail pass still on this one. I think I do. at
Yeah.
least.
Okay. Well, we'll see that. Yeah. We'll look at that. That.
It's the version of FastQ we're using, but that's okay. There's several, FastQC, I'm sorry, there's different versions, but that's something we'll work on. Okay, so just make a
Yeah.
note of it.
Okay.
So,
Yep.
So my question, I guess, would be, it's a different version, so it's giving us somewhat different information, I presume.
I think so.
Can you look at this and tell us whether this information is useful or how to interpret it?
I don't think it's giving you I think, let me qualify that, not different information. There's just a couple of new reports that they've added to the newest version
Probably. Yeah.
of FastQC.
I guess my question is, we've got something different than you've got.
Are you
Can
clicking?
tell us
No,
what
you
it
have,
means?
which one are you missing? You have this.
No. So they have. But they have.
It's truncated at 10 plus and it's missing the blue line.
Right, well,
Yeah,
let's
but
just
you
figure it out.
you do have sequence duplication level report, is you have that.
So
But I'm just asking what, and it's showing a fail, so, or an X, so I'm asking how can I interpret that?
Right, we will go through.
yeah. And let's the next plot is also.
Sorry.
Okay, go on.
There's a number on top that says sequence duplication level greater than 92.5%. Does that?
Sequence duplication level.
Yes.
And mine is greater
Equal
than
or greater than
56%.
92.
Yeah, mine's like 58%.
Does that mean anything?
Yes, it does, but we're gonna go through.
Okay.
Okay, we're
Yeah,
gonna do
and
that.
what we should do is,
Yeah.
I
Yeah.
think let Nicolas go through,
I-
because
I-
everybody, this is the point.
I'm going to
Everybody's
finish.
gonna have a different
Yeah.
thing, so we're gonna go through.
So the other way to look at duplication in a way is the K-mer content. What is this? Oh, okay, it didn't show up. So I think on the DNA Studio you have something about K-mer content, and that gives you the most abundant K-mer. So K-mer are just short fragments. So it scans the entire FastQ and look at which ones are the most abundant, and try to make an estimate of if that's a reason to worry. Is there something that's more abundant than we expect or not? So I don't know, maybe some of your report might, I don't know if they report something wrong with that, or do you have that on your DNA server?
Yeah.
Okay. So again, we'll look at that. The last thing I want to mention was to get back to this image with the fragment and the sequence. This is all fine when we have a long fragment and we sequence the extremity. But what we can also have, so basically it's fine when the fragment length, the blue line, is greater than twice the read length. Okay, so that's those cases that we have, and talk about that later. But in some cases we have the fragment length that's shorter than twice the read length. In those cases, the fragment here that we're going to sequence are going to overlap. Okay, so the 3' end of your read 1 will be the same as the 3' end of your read 2. Okay, and when the fragment length is really shorter than the read length, you will start to sequence the adapter sequence. Okay, so I didn't illustrate it but you have adapter sequence, and they will be part of your reads. Okay, so when you have those kind of situation, it's good to do what we call adapter trimming. And so when the fragment size is lower than the read size, you'll sequence part of that. And one tool that I think is presented in DNA Subway and was developed here at Cold Spring Harbor is this thing called FastQ Toolkit, which lets you input the sequence of the adapter. It will find that sequence in all of the reads and remove the 3' end part that match to the adapter. Okay, so what you end up with is a FastQ files of different read lengths. Okay, because every time it will remove the part that match to the adapter in the 3' end region. It's also possible to do quality filtering, and so to remove all the reads that don't match a certain quality threshold. And so yeah, here we are. So in summary, the QC check, you want to check that your file is the expected size. And so I saw when I arrived that most of the files were in the 1 gigabyte range, which is probably what you kind of expect. You want to check what's the number of reads. Okay, so it will never be the same number, but you want to have a good number, the number you paid for usually. Well, which no, but it's one of our deliverable. When we do a project, we say we're going to give you FastQ files and we're going to sequence 10 million reads or 20 million reads or 30 million reads. Okay, in the exome world, the deliverable is we're going to give you 30x coverage or 40x coverage or 80x coverage. So each location of the genome will be covered by 80 reads. Okay. That's kind of a different metrics, a different parameter in the experiments. So you want to check that you have the right number of reads. You want to check that it's the right read lengths. They're all the same read lengths. The composition of the sequence, that you have a consistent quality along the sequence, that you have a low duplication rate, and we'll go through that. That you have a good diversity of the library, so that you don't have you didn't sequence always the same one. And in RNA-seq and mostly in small RNA-seq, that can be a problem because in microRNA can be extremely abundant, and it's very frequent that 80% of your library is matching to 5 microRNA. Okay, so you try to sequence 1,000 microRNA, but because it's a quantitative experiment, a lot of your library will match to the same sequences. Okay, so if you want to discover new poorly expressed microRNA, you'd have to sequence a lot of reads in order to get those small ones. Okay, so the the curve of the abundance is really a long tail shape, if you want. And so you don't want to sequence the adapter sequence, or if you do, you want to remove them. And still, yeah, medicine can go wrong. So even if you're fine with all those steps, you're not done. And so you need to map the reads and assemble them into transcript and quantify the transcript. And that's what we'll do after lunch. So if you have any question now, I'm happy to answer. Thanks. I think I'm done. Yep. Yep.
I just have a question. Sorry. I was just looking at the K-mer graph. And I was wondering if you could, I mean, I'm sort of getting it, like we got a little 5-mer and if it's overrepresented.
Can I look at it?
Right at the beginning?
Yep.
Yeah. Because I think
Okay.
other people probably
Yeah.
look similar to mine.
Yep.
Yeah.
Maybe what would be useful is if you put your laptop on the projector, Judy.
Or maybe you can just draw it. You can draw it
Or
using
you
your
could just think about it, because I'm sure other people's look like mine.
Yeah, that's what I'm thinking. Maybe if we go through an example.
Okay.
Not necessarily perfect data, but data that you can then filter and trim appropriately. We're all going to have to go through the
I think a lot of
process
people are going to have a
trying
similar
to,
type pattern.
trying to
Sure.
make some decisions. And while that's being set up, let me tell you about the next option that you have. So there, the next thing to do in DNA Subway is the FastX toolkit, where you then go through the process
You shouldn't
of
have
trying
to,
to
you
take
should
reads
just go in.
and either trimming or filtering or whatever it is. And you'll notice that there are two options there. One is called basic and one is called advanced. So the basic one option that you'll have will give you the option to choose some reasonable defaults. If you don't want to think, you can try that and see what happens and then look at the QC report again and see how much it improved. But once you really do understand what's going on here, you can then try to adjust those settings and those parameters to give you the best type of cutting and filtering given your data. And then I also mentioned, and maybe you want to do Control 0 to zoom out a little bit, or Control minus, because there we go. I want to also mention, so obviously you've also probably gotten the idea that things are either green check or warning or fail. And just because you have a fail doesn't necessarily mean you can't go on, but some fails are worse than others, and as we go through, we can take a look. So I'll let Nick kind of, yeah.
Sorry. So in anticipation of using this as the primary context or vehicle for
Okay.
If you scroll down, you
Are
see
there-
like K-mers.
Are there or will there be help screens that answer many of the same questions that came up?
So this is a good point. So at the moment, there is not any particular help attached to this as far as I know. Yes?
There is.
Or is there a, where is it?
So what exactly?
Well, when we're help.
Like help screen of.
Yeah, when
what-
all the questions that
Yeah,
came up. and the
but I'm just
questions-
saying, so what is the solution at the moment?
So there is some help built into DNA Subway, and I'll show you that. But for now, if you're talking about analyzing these graphs,
Well, the deduplication content, or some of the parameters and the data, the shape and what these scores mean and things
Okay.
like
So
that.
Can you show us what help is now?
so I can
Yeah,
show you
yeah.
what help is now. But for that, the best place to look on that would be the documentation page for the FastQC.
But
like, you know,
assuming that nobody knows that, I'm just saying,
the actual tool.
But there's no context-specific help
Well,
available to you.
There is a little bit of help. We'll work on making this as good as possible. So in most windows, if you just, sorry I did that too fast maybe, but in most windows there's this icon at the top right of the screen, this help icon. If you just hover over that, it will give you some information about the tool that's being run there. So there's a bunch of information here, and then there's also a link to the actual tool itself. Now this is for the FastX toolkit, which we'll be looking at next. For FastQC, I don't have that help up for the tool itself yet. However, things like what the graphs mean, what's a good graph and what's a bad graph, all that information is available on the tool page for FastQC. I wouldn't want to necessarily bring all that information into Subway if it's already available.
It's linked on the wiki, the FastQC documentation.
Sure.
So it links to that first website here, and then if you scroll down a little bit, there is, but that aside, that being said, things like contextual help, I think that's your job as a group to also provide recommendations of things that you'd like to see.
That's the
And
report
for
that
us
I
to
showed.
see
So.
what we can actually
Yeah,
implement.
yeah.
And Agnes?
But
So
I,
is there
I,
a link here to talk about
I think there is.
PDF
what
file
each
of the
these-
FastQC report so that
So,
we can download
I
that?
think, do these export as HTML, Mohammed, when they run? Or
Does-
I mean, when it's done? Or is it somewhere on your storage now?
It is in the storage, so if you wanted to have that on your disk, you can acquire that from the DE, and I can show you more about that. Yeah.
Yeah, so there's some things have automatically an export feature where you can export the data or at least point to where it is in the discovery environment. But that is an implementable feature, for
There's a
instance.
video.
You know, export PDF type of thing.
Okay, so what I see in DE under my data is there's actually.
It's not, I don't think it's with your data.
Oh, it's not
but,
with the data?
Yeah, it's somewhere else.
But all of the tools which run in DNA Subway, all of the output is available in the DE, all of it. So we don't necessarily show all of it.
Somewhere they have,
in Subway,
Some examples.
but all the output.
Yeah, I don't have it. They have a
that
help
does
actually,
produce is
like that
available.
explains
So
those
the
things.
help
And I'll show you that.
is not here. Why would it be where you think it is?
And this green line, you guys, is really beta
It's
right now, so
actually,
we're really counting on you.
well, the one that I goes to is here, and it's in these directories.
Oh yeah,
And then
okay.
if you go to like analysis modules,
I got it.
and then I think they at least tell you what is a
Yeah.
fail and what is a pass.
Oh yeah, okay.
Yeah.
Cool. Yeah. Oh yeah, actually.
So that's in that directory.
So in the help actually.
Yeah.
Okay.
So if you want, so as an aside or as a supplement, if you go back, so on the wiki, I'm sure by now you've found it, or if you just Google FastQC, and then we'll maybe go to, actually the reason why we have Judy's computer up here. But if you go back to, on the wiki for today, it says FastQC documentation, and it'll take you to this page, or if you Google FastQC, this page from Birmingham will be the first thing you find. So further down, there's this documentation, and it says a copy of the FastQC documentation. So if you click on that, and it's not in the most friendly format necessarily, but down here, the one that I always refer to is analysis modules. And then if you click on any one of these, it goes through a summary of what that shows, and then gives you some explanations. This box is this, or this is the quartile, or this is this, or this is the access, and it tells you what is a warning, what is a fail, and maybe some reasons if they, have an idea of what might be behind that. But I think we were going to try to go through maybe Judy's as an example of real data, and she has some reads, and we wanted to take a look at that. So I'll open that back up, and then maybe we can try to take a look
Do some magic.
and see what we see here. And I've seen on your reads and somebody else's, this first thing, if we scroll down a little bit, on the per base sequence quality, I see with a number of, well, 2 observations, that you guys have the first base or 2 with a little bit lower quality. It's not horrible at all, but it's a little bit lower. Okay. It's not perfect.
So.
And sometimes there's not a 100% true. This must be why. There are lots of reasons why things could be one way or another.
This is
To try to guess it at that, you can sometimes. But remember, also this is a new machine, NextSeq. So only a few people have that, and every machine I think has its little idiosyncrasies. I'll just let Nicolas explain because he's the expert on that kind of me.
Yeah, but I'm trying to come up with good explanations.
He has to think a little bit on it.
So, this, we were talking about the FastX. Should we stop running that, or?
I don't think you should run FastX necessarily until you've had a time to try to assimilate and understand some of the things. You can run it and use the basic mode, and it will do some filtering. To me, and I don't know how many people's data this looks like, they said maybe you need to have some time to look at each other's.
Are other people's
To
looking
me, what I've
at
seen a couple of people is that the first few base pairs,
Yeah.
we're getting some sort of lower quality. I mean, traditional
But-
Illumina, I think we expect the last few base pairs, you get that, and you may have that too, but I'm getting the first.
But don't you have like a lower diversity on the first base also? Because you know, one element that I didn't talk about is that a lot of those QC are related one to each other, right?
Well, for sure, the like per base sequence content and the per base GC content are totally related to.
Yeah, of course. But even there, the base composition along the read might have something to do with. So first of all, with the K-mer content, okay, right? So what we see here is like overrepresentation of some K-mers in the first bases, and that's very correlated with that one, okay? But it might also be correlated with that one, right? So here we have I don't know, there seems to be something going on in the first 10 base. Yeah, one thing to mention here on this FastQC is also that you have the first 10 base, and then they're grouped by some bins. So it's 10 to 14, 20 to 24. Okay, so they're a group of 5. Okay, which might flatten the lines also a little bit. Okay, if you have this if you take the blue lines over the first 5 bases, you probably have a flat line that's at the same level as those ones. See what I mean?
Yeah,
right?
Yeah, no, I do.
So.
But shouldn't that be taken into when it fails and passes, or I guess whatever?
Well, so here it failed on this one. So I think, in my opinion, that's a limitation of the FastQC, that they do this binning here after 10 base. Sometime I would like to see the entire thing, and I would compute it myself. So.
Because that could explain that would exactly explain the K-mer thing.
Yeah, but I'm wondering if it's also related to the sequence duplication. Like, if you have.
The quality score at the top.
If you have really one read that's super abundant, and say that's a C, T, C, A, so you can basically read the that would be possible. Then you have some organism have a very abundant, or some organism and some tissues and some cell type have a very abundant transcript. If you do blood RNA-seq, if you don't look carefully, you'll sequence a lot of hemoglobin. Right?
Plant sequence a lot of
So I don't know exactly what you did, but it might be that.
It's leaf tissue, so you could sequence a lot of Rubisco.
Right. But then, the other thing is, if it's a long transcript, you would expect, sampling of the entire transcript. So my point was more like for short RNA, where you would systematically do the same thing. But
right, it wouldn't. Right, if it was.
Yeah, I wonder if here, and that's something we will see once we do the gene expression, that maybe there will be like one super abundant transcript that kind of bias all the different metrics. But it's still, with that QC, you definitely go to mapping, to quantification. You just keep somewhere in your head that you have those information, and they might relate to something you observe.
Like trimming them off at this point.
No, I wouldn't see a reason why. And really, usually trimming is on the 5' end. There is no reason really to trim the 3' end. I mean, clearly this is very good. This is very good data. I would go for that.
In a sense, when you're thinking about trimming, you kind of use this first graph.
Yes.
Okay.
Yes.
So that's why you shouldn't get alarmed necessarily because something failed. You really have to consider.
Yeah, no. So those calls here, I mean, I don't know anybody who really takes them at face value.
Okay.
I'm not even sure why they do that.
It's comforting.
I mean.
No, but well, except when it fails.
Well, if you got it right.
When it's green, it's fine. But when it, so yeah, if I was doing my own software, I wouldn't make the calls. I would just give the values. Yes.
So if you have, like, if you fail at sequence duplication levels, is there a way, or should we deduplicate?
Yeah.
Somehow? How can we do that? Because I don't think FastX actually does that.
No, I don't think it does, although I don't think so. There is a way to do it. Shall we do deduplication? If you have, gross duplication. Well, I mean, over PCR amplification. I don't know which tool can do that in DNA Subway.
So in DNA, I don't think you can do that. I think maybe somewhere downstream, like in Top Hat, I think you can provide like some kind of mask file to not align. So it's not really set up to do that.
That's in cufflinks. You have the mask file to not quantify certain transcript.
Yeah, I think you can. So I think you can, like, let's say you have ribosomal RNAs,
Yep.
things that you know that you're not concerned about. I believe you can provide, and I don't know if you
Yep.
can do it in D-Subway, but I think in Top Hat you can provide a mask file to tell it not to worry about certain things.
I think it's cufflink.
It may be.
I think it's in the quantification.
Yeah.
What if I just want to find the reads here that start at the same position and end at the same position, and I'm going to assume that they're all coming from my PCR application that was, I don't know, too many cycles, and I just want to get rid of them. Can I do
So
that?
here, when you mean location on the genome, or?
Well, if I have two reads that are identical, that
Yes.
start like in exactly the same place, and I'm
Well, they have the same sequence. Yep.
Right? They're
Yep.
just identical reads.
Yep.
Should I get rid of
So
those extra ones?
once again, I don't know enough about DNA, so I don't think you can do it.
But why would you want to get rid of duplicate reads?
Well, because I guess duplicate reads are bad, right? Because if there are some regions that are over
So-
behind your PCR.
So they're not systematically bad. So for highly abundant transcript, you expect to have some duplication, right? So it's a sampling process, okay? So you have, if your transcript is like 1,000 nucleotide, okay? If you have 1,000 reads, you would expect them to sequence at every location. If you have more than 1,000 reads, they would start to start at the same location.
Yeah, but they won't necessarily start and end at the same place, right?
Right. But they're all the same size. All the reads are the same size. So here I'm talking about single end, but even with paired end, if you really have a high level of expression, by chance, you'd have reads that are at the same location. Now, so that's kind of the reason why we don't remove duplicates. And very frequently, you have one transcript that's several percent of the library, right? 5%. 5% of it's very frequent that 5% of the library of the reads that map to genes are mapping to one single gene. Okay, that's your most highly abundant transcript. It's very frequent, and so that can be millions of reads actually. Okay, so if you remove the duplicates, you will underestimate the expression of that gene.
Isn't that if you were to deduplicate?
Yep.
Wouldn't you be defeating the entire purpose of the RNA-seq analysis, which is to compare relative transcript abundance?
So it's kind of a debate. I mean, what I describe here is like if you have two reads, right? Now if you have 50 reads that are all identical, it's very unlikely that it's by chance. It's most likely because of a PCR artifact. But at the moment, if 50 reads are all identical, like paired, and they're
Why isn't that just the same as 50x coverage is the aggregate coverage?
Yes.
that's the aggregate over all the reads. Yeah. That's per location here. How many reads? Yep.
Because you don't expect that they will, by chance, line up end to end.
I'm talking about 50 identical reads, both R1 and R2 are identical. That's most probably a PCR artifact.
You jumped now to quantification, right, or at least to the mapping, because you have overlapping non-identical reads indicated in your
Yes.
right?
Yeah.
He's saying if
So
they're
right
example-
now with the FastQC, we're only talking about analyzing individual.
No. Yeah, exactly. No, I'm going here at the very end of this afternoon's talk, which is the quantification. But that's what matters, right? That's the end goal, is to have the best possible quantification of the genes based on the reads that we sequence. Okay? And so what can we do along the process to make sure that the quantification will be the best possible? And shall we remove duplicate reads? The consensus at the moment is not. And I don't think I saw like a very clear, perfect paper that described different ways of doing it. I mean, maybe that's actually, now that I think about it, that's something we could do, and to, maybe keep reads that are in two copies and remove the one that are in 50 copies. That's actually something that makes sense. But the effect on expression will be probably very located on a very few genes. Okay, so there might be a few genes that are overestimated, on which the expression is overestimated because of that problem. Usually we try to control the duplication level both at in the FastQs with FastQC that I just showed, and after mapping, and I'll talk about that after. Okay. Yep.
What do we do with the overrepresented duplicates? The overrepresented sequences or reads?
The reads or the K-mer? The reads, that's essentially what we described. So at the moment, I say mostly people keep them, try to keep everything. The mapping step will remove the very low quality reads. Okay, the reads that are very bad quality, they won't map the genome most probably, so they will be removed. And the reads that are in multiple copies, at the moment the general consensus is really to keep them. If I find time, I might study that and what could be different strategy to sort this out. But at the moment, the consensus, and I think that's what you should teach,